Lorraine, did you want me to wait for another minute before we begin?
Yes, please.
Okay, you just tell me when to start.
Okay, let's go ahead and get started.
All right. Let's see. Hello, everyone, and thank you for joining us today. We're pleased to announce two speakers. We'll introduce the next slide. This is a webinar series sponsored by the American Chemical Society, ACS Infectious Diseases. And this is a journal. I have a few points on this for some of the viewers who may not be familiar. We're a relatively new journal launched in 2015. We really pride ourselves as the first chemistry journal that focuses on infectious disease research. And we have a pretty broad scope, highlighted some of the topics we cover below, but really the underlying theme of the interest for us is chemistry, using chemistry to study infectious diseases, whether it's pathogens or host pathogen interactions, development of therapeutics with an emphasis on mechanism, the development of vaccines using novel chemical approaches, or diagnostics and drug delivery systems, again, using innovative and novel methods. So let's move on to the next slide. Okay, so I'll hand it over to my colleague, Peter Tong. He conceived this webinar Zoom series. So please, Pete.
Yeah, thanks, Courtney. So the genesis of this meeting was during the pandemic when nobody could go to conferences, and we were very concerned about the development of young scientists and their ability to meet other people and share their scientific ideas and basically participate in the scientific community. And so we started this pop-up Zoom meeting as a mechanism to bring together scientists from across the world and also give a platform for junior scientists to present their research. And so today we have two speakers from India, Geetika Dhanda from JNCASR and Mangal Singh from IIT Roorkee. And they're both going to give talks today, and the first speaker is Geetika.
Yes, and before we begin, I just wanted to remind the viewers that you can ask questions in the chat or Q&A function. Please use those, and at the end of each talk, we'll then, Pete and I will both serve as moderators and ask questions on your behalf. So thank you.
Thank you, Professor Pete and Professor Courtney. So I'll just share my slides. I hope my slides are visible. So hello everyone. The title of my talk today is Small Molecular Adjuvants, Resistance Breakers that Revitalize Obsolete Antibiotics Against Gram-Negative Superbugs. So before going into the details of today's discussion, let's look at the ruckus which antimicrobial resistance has created globally. The annual global deaths predicted due to AMR by 2050 are expected to be 10 million, and which will put a major problem on the healthcare costs, amounting to about $100 trillion, which is going to be a severe problem for us if the problem is left unchecked. And if you look at the backdrop of this, the number of antibiotics which have been approved over the last few decades have decreased considerably, which means that there is a huge gap between the availability of new antibiotics and the immediate need to combat antimicrobial resistance. Thus, it's safe to say that there is a pertinent need to develop novel strategies to combat the most critical pathogens. And currently, as of 2020, we have around 400 plus candidates in the global antibiotic preclinical pipeline from about 300 institutions in the world. And most of these focus on infections with gram-negative bacteria. Now, why is this the case? So before I answer this question myself, I would like to hand it over to Lorraine to ask the first poll question for today's webinar.
Yes, thank you. So we would like to invite the audience to participate in a poll question that I'm going to pull up right now. Okay, and it's which is the top most critical priority pathogen according to the World Health Organization? Is it MRSA, Neisseria gonorrhoeae, Acinetobacter baumannii, or Helicobacter pylori? Okay, we'll give everybody a few seconds to respond. I see the responses are coming in. Looks like there's 2 runaway answers here. And we'll give everyone 10 more seconds. All right, I'm going to close the poll in 3, 2, 1, and here are the results. Back to you, Geetika.
Okay, thank you, Lorraine. So I can see that there is a tie between MRSA and Acinetobacter baumannii. So let us look at the priority pathogen list, which was, just a second, I cannot move forward. Yeah. So let us look at the global priority pathogen list, which was published by the World Health Organization in 2017. And according to that, Acinetobacter baumannii, which is resistant to carbapenems, is the most critical priority pathogen. And in this list, as you can see, that all pathogens of concern have been divided into specific priorities like critical, high, and medium. And in the critical priority pathogen list, we have gram-negative bacteria like Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacteriaceae. And all of these bacteria are resistant to carbapenems, which is one of the last resort antibiotic for treating gram-negative bacteria. So now if we look at the approaches to tackle this problem, there have been various efforts all over the globe. And one is definitely the development of new antibacterial agents or drugs and identification of novel targets. And in this approach, the bacterial membrane has proved to be a good target, wherein antimicrobial peptides, pre-existing antimicrobial peptides, and cationic hydrophobic small molecules as well as polymers have been designed to target this membrane. Now, another approach, which is slightly non-conventional and which is the focus of today's webinar, is the rehabilitation or repurposing of approved drugs by using molecules, adjuvants, in combination with obsolete antibiotics, which can improve the activity of the antibiotic. So before I go into the details of how to design these adjuvants to target certain pathogens, let me focus on this critical priority pathogens, which are the gram-negative superbugs, and let us see what makes these bacteria attain the superbug status. As you can see that for gram-negative superbugs, there are very less number of treatment options, and they show resistance to almost all antibiotics. In this graph, you can see that only tigecycline and colistin are active against these bacteria, and reports have surfaced which show that even these 2 antibiotics have become ineffective against certain strains of gram-negative bacteria. Now, if to dwell into this, that why these gram-negative bacteria have attained a resistance to a number of antibiotics, we have to look into their structure and physiology. So in these superbugs, there are a lot of enzymes like the beta-lactamases, which degrade certain antibiotics like beta-lactam antibiotics and thus reduce their efficacy. So this is one resistance mechanism which is prevalent in these gram-negative superbugs. Another is the presence of these multi-drug resistant efflux pumps, which continuously extrude the antibiotics outside of the bacterial cell, thus reducing their intracellular concentration and reducing their activity. And the functioning of these efflux pumps depends upon the membrane potential or delta psi. And another intrinsic resistant element which is seen in gram-negative bacteria is the presence of an outer membrane, as you can see over here, which is like a permeability barrier to a lot of antibiotics. So These three resistance mechanisms are ubiquitously found in a lot of gram-negative superbugs. And as you can see in this table, various classes of antibiotics like tetracyclines, rifampicin, chloramphenicol, beta-lactams are rendered ineffective due to these resistance mechanisms. So what if we devise a strategy to counter these resistance mechanisms? So if we have a molecule which will counter these resistance mechanisms, and if that molecule is used in combination with the appropriate antibiotic, then that therapy would be a suitable therapy for treating gram-negative superbugs. So this brings me to the topic of today's discussion, which is antibiotic adjuvants. Antibiotic adjuvants are compounds that have little or no antibiotic activity themselves, but they act to block resistance or otherwise enhance antibiotic action. And these adjuvants can broadly be classified into class 1 and class 2 adjuvants. Let us look at some examples of these adjuvants one by one. So class 1 adjuvants can be further classified into class 1A adjuvants, which directly inhibit antibiotic resistance elements. Like an example of this is clavulanic acid, which is an approved serine beta-lactamase inhibitor. It directly stops the functioning of beta-lactamase enzyme, which renders beta-lactam antibiotics ineffective. Another example is class 1B adjuvant, which passively target resistance elements, like they circumvent resistance mechanisms, including metabolic pathways or physiology, which indirectly is able to counter resistance seen in these superbugs. An example of this is lopramide, which affects the membrane potential of gram-negative bacteria. And because of this, a number of tetracycline antibiotics, the influx of tetracycline antibiotics can be increased. Another example of these adjuvants is class 2 adjuvants, which do not act on bacteria at all, but they act on host properties to enhance antibiotic action. An example of this is streptozolin, which recruits more and more macrophages to clear infection in our body. Now, although a lot has been known about these different classes of adjuvants, but the adjuvants seen in the market currently are only class 1A type adjuvants. And these also are restricted to serine beta-lactamase inhibitors. Some examples of this is tazobactam, avibactam, gabapentin, and relebactam, which have been approved by the FDA, and they are used with their appropriate partner beta-lactam antibiotics. So this means that a lot of work still needs to be done for translation of class 1B and class 2 adjuvants in the market. Now, one class 1B adjuvant, which is in clinical trials currently, is SPR 741, and this is by the Spero Therapeutics. It has been derived from a known antibiotic polymyxin B, and this basically perturbs the outer membrane of gram-negative bacteria by interacting with LPS. And because of this, it potentiates a number of antibiotics which are resistant because of this mechanism. So here is an example shown, the combination of SPR 741 with rifampicin. And you can see over here that when the efficacy of this was checked in a lung infection model of A. baumannii, which is the most critical priority pathogen, the combination indeed reduced the bacterial load as compared to the untreated and individual adjuvant and antibiotic control. But apart from this, no other class 1B adjuvants are in clinical trials. So this brings me to our approach of tackling this problem of gram-negative superbugs. If we look at the membrane of these gram-negative bacteria, they comprise of various negatively charged lipids, like lipid A, which is a constituent of the LPS, phosphatidylglycerol, and cardiolipin, which are negatively charged. And obviously, they have lipophilic moieties, as you can see over here. So what if we design an adjuvant which is positively charged in nature and which is amphiphilic, so that it could interact with these lipids present in the membrane of bacteria, and such that it is non-antibacterial in nature, but just weakly perturbs the membrane. And this weak perturbation will basically counter resistance due to membrane-associated resistance elements, which we have already discussed in the earlier slides, like impermeability and efflux. So because of this strategy, if you look at it, some antibiotic, which is obsolete to treat these gram-negative superbugs, suppose antibiotic A, it is not able to act against bacteria. So we have live bacteria. The adjuvant just weakly perturbs the membrane. So again, it will be non-antibacterial in nature, and the bacteria will be alive in this case also. But when we use a combination of the antibiotic and the adjuvant, because the adjuvant is targeting these membrane-associated resistance elements, which render this antibiotic ineffective, the combination would actually work and this would kill the bacteria. So this is our strategy in general, but to design such an adjuvant, which will be non-antibacterial in nature, which will be non-toxic in nature, and also it should weakly perturb the membrane, we need to optimize certain design parameters in molecules. So for this, we have designed certain molecules consisting of positive charges, because obviously we need positive charge to interact with the negatively charged bacterial membrane. And we have varied the hydrophobicity by acyclic aliphatic, as you can see in the long chains here, or cyclic aliphatic or aromatic hydrophobicity. And when we observed the membrane perturbing properties of these different classes of adjuvants, and it was compared with a known surfactant molecule, CTAP, we could see that they could be characterized into 3 classes of membrane perturbing compounds. CTAB, obviously, because it is a membrane disruptor, it was highly membrane perturbing in nature, as it caused high outer membrane permeabilization and membrane depolarization. Whereas the acyclic aliphatic molecules, they were moderately membrane perturbing in nature. And we observed that in going from the acyclic aliphatic to the cyclic moieties, or the cyclic hydrophobic molecules, the extent of membrane perturbation decreased considerably. So to further validate the difference between these moderate and weak membrane perturbing compounds, we studied the leakage of a dye called carboxyfluorescein through liposome mimics of bacterial and mammalian membrane. And if the membrane is perturbed to a great extent, then this dye would leak out and it would show fluorescence at a lambda max of 517 nanometers. So over here, you can see that CTAP was able to cause a high dye leakage, and even our acyclic aliphatic compound like NC12 was able to cause some amount of dye leakage in both the membrane mimics, whereas our weak membrane perturbing adjuvants, that is NADA and N-diethy, they didn't cause any leakage of the dye, which confirms their weak membrane perturbing nature. Now, further, we wanted to check the antibacterial activity and toxicity of these different classes of compounds. And it could be observed that the surfactant molecule, which was high membrane perturbing in nature, it showed high activity and high toxicity. The compounds with acyclic hydrophobicity, they showed less activity, but definitely high toxicity, as can be seen by these low EC50 and NC50 values. But the compounds with cyclic hydrophobicity, or our weak membrane perturbers, they didn't show any antibacterial activity, nor did it show any toxicity. So we have concluded from here that by incorporating this cyclic hydrophobic moiety, the molecules were weak membrane perturbing, non-active and non-toxic in nature. Further, we wanted to check whether this weak membrane perturbation is enough to potentiate antibiotics. So we have taken 3 different classes of antibiotics, which are ineffective due to membrane-associated resistance elements like efflux and impermeability. And we have checked the activity or the MIC of these antibiotics in presence and absence of the adjuvant. And you can see that even in the presence of the cyclic hydrophobic adjuvants, the MIC was reduced, which means that cyclic hydrophobicity in adjuvants is sufficient to potentiate antibiotics. So with this work, we have optimized the design parameters of the adjuvant that we desired. That means non-antibacterial, non-toxic, and weak membrane perturbing in nature. So it should comprise of certain positive charges and a cyclic hydrophobic group. Further, we wanted to check the spectrum of potentiation of this adjuvant, where we have taken clinical isolates of gram-negative superbugs, including carbapenem-resistant strains, and also the beta-lactamase producing strains, against which there is no approved antibiotic. So you can see that potentiation factors from 8 to 4,000 fold were observed for all these classes of antibiotics in presence of the adjuvant. And further, we wanted to check that whether other antibiotics, which are ineffective due to membrane-associated resistance elements like doxycycline, tetracycline, chloramphenicol, and vancomycin, were they potentiated by our adjuvant. So indeed, by performing checkerboard assays, we could observe that the light color here represents no bacteria. And in the presence of the adjuvant, you can see that there is no bacteria at 1.12 microgram per ml of doxycycline, which means that the adjuvant with cyclic hydrophobicity exhibited a broad spectrum of potentiation with various antibiotics and against all critical gram-negative pathogens. But after optimizing the parameters, we observed that this adjuvant actually needed high concentrations of 64 to 128 microgram per ml to show potentiation of various antibiotics. So we wanted to, and usually these concentrations are difficult to maintain physiologically. So we wanted to slightly improve the design by keeping the cyclic hydrophobicity constant, but increasing the backbone hydrophobicity and also the number of positive charges from 2 to 4. And this was our lead compound, which we obtained. And this compound also was non-toxic in nature and non-antibacterial as well, as can be seen from these EC50 and HC50 values. And indeed, by changing this slight design at much lower concentration of 8 microgram per ml, the adjuvant was able to kill the bacteria in presence of the antibiotic. As can be seen from here, there is no bacteria present when it is treated with the combination. Then we have checked the MIC of 3 different classes of antibiotics in presence of our adjuvant against the most critical priority pathogen, which is A. baumannii. And you can see that the MIC or the inhibitory concentration to kill the bacteria was reduced from 64 to less than 0.125 to 1 microgram per ml. And further, with the lead compound, we have checked activity against critical gram-negative superbugs, including carbapenem-resistant strains, and all of these are clinical isolates. And we have plotted the FICI value. So this is the fractional inhibitory concentration index, and a value of less than equal to 0.5 indicates good synergy and potentiation. And you can see from here that against all these antibiotics, with all these antibiotics, the FICI values against all these bacteria was less than equal to 0.5, which means that our adjuvant was indeed broad spectrum in nature, and it could combat a number of clinical isolates of gram-negative superbugs. Then we wanted to further investigate the mechanism of potentiation of these antibiotics, of these adjuvants, sorry. And as mentioned earlier, we have used these fluorescent probes to investigate outer membrane permeabilization and membrane depolarization. And you can see that our adjuvant was able to cause these slight perturbations in the membrane of bacteria. And it was much less as compared to the surfactant control CTAC. Then we further wanted to see that what is the effect of this membrane perturbation on the delta psi or membrane potential of bacteria. So we have taken another dye, whose monomer is green, known to fluoresce green in color. And if the delta psi of the bacteria is highly negative, which is in the real case scenario, it forms red aggregates. But if the delta psi is decreased, then these red aggregates would reduce. So you can see in the untreated control, a number of bacterial cells show the presence of these red aggregates, whereas when our compound adjuvant was treated, the number reduced drastically, which means that our adjuvant reduced the membrane potential of these bacteria. Now, as I have mentioned in the earlier slides, there are a lot of efflux machinery which depend on the bacterial membrane potential. So we wanted to see, due to this decrease or due to this perturbation, is the efflux of antibiotics affected or not? So we have taken a fluorescent substrate of ACR-AB Toll-C efflux pumps in gram-negative bacteria, which is ethidium bromide. And if the efflux machinery is in bacteria is working, then this ethidium bromide would be continuously extruding out, and the number of cells which stain positive for ETBR would reduce. So you can see from the shift from here in this graph that in the untreated case, the number of cells which stain positive for ETBR were quite less. But if the efflux machinery is hampered, then this shift won't be observed. So when we have treated the bacteria with our compound, then the ETBR, the number of cells staining positive for ETBR is maintained, which means that our adjuvant hampered the efflux machinery. So in conclusion, our adjuvant permeabilized the outer membrane, depolarized the inner membrane by decreasing its membrane potential, and inhibited efflux, thus countering resistance elements like a class 1B adjuvant. Now, after investigating the activity of our adjuvant against a panel of gram-negative superbugs and also investigating its membrane mechanism of potentiation, we wanted to see whether it is able to tackle more complicated forms of infections like bacterial biofilms. So these biofilms are formed by bacteria which are in different phases, and they can assemble on biotic or abiotic surfaces and form a matrix around them comprising of DNA and polysaccharides. And this acts as an impenetrable barrier for the action of various antibiotics. And these bacterial biofilms are relevant in a number of diseases like cystic fibrosis and other associated respiratory infections. So it is definitely important for us to develop a strategy to combat these bacterial biofilms as well. So we have developed biofilms of DS-red-tagged P. aeruginosa on glass cover slips. And we wanted to see whether our combination of adjuvant and antibiotic was able to disrupt these biofilms or not. So you can see over here, like in the untreated case and even in the minocycline, fusidic acid or individual adjuvant treated case, the biofilms were intact and they flourished also, which means that the bacteria and the biofilms were live. But when we use the antibiotics in combination with our adjuvant, the thickness has considerably reduced and the fluorescence intensity has also decreased, meaning that the bacteria are dead in these biofilms. Further, like we wanted to see whether this approach would be translatable in a real case scenario. So like I mentioned earlier, in respiratory infections, usually they are associated with mixed species biofilms of P. aeruginosa with MRSA. And even though there are antibiotics to treat both of these bacteria and MRSA specifically, that those cannot penetrate the biofilms. And because of this, the disease case scenario is worsened. So because of, for this, we have created mixed species biofilms of P. aeruginosa and MRSA. And we wanted to see whether our combination of adjuvant and antibiotic could reduce the biofilm embedded bacteria as well as the dispersed bacteria. So indeed, the combination showed a reduction in the bacterial load in case of biofilm as well as dispersed bacteria as well. And this was not seen with a known gram-negative antibiotic colistin, which means that this is a severe problem and we have been able to tackle this through our combination of adjuvant and antibiotic. So in conclusion, the biofilms of P. aeruginosa and mixed biofilms of P. aeruginosa and MRSA were eradicated. And also the dispersed cells of biofilms were inactivated by combination of antibiotic and the adjuvant. Further, we wanted to see whether our combination of adjuvant and antibiotic works against another phenotypic resistance element, which is intracellular infections. So certain bacteria like P. aeruginosa are known to enter the host cells and form an endosome. And because of this, they would evade the immune mechanisms of our body as well as the action of antibiotics. And because of this intracellular infections, there is relapse seen in a number of infections, which is difficult to treat. So we have to see whether our combination works against these intracellular infections. We developed an intracellular P. aeruginosa infection model in raw macrophages. And after 24 hours, we have lysed the cells and seen the bacterial load. And you can see that in the combination of minocycline and our compound LB-DIFE, around 5 log reduction in the bacterial load was seen. And even the microscopic images proved that like at the end of 24 hours in the untreated case, the cells were completely harmed and lysed by the bacteria inside, and all you can see are debris. Whereas when our treatment of minocycline plus LB-DIFE was given, the morphology of the cells is maintained. So the adjuvant was indeed effective in reducing intracellular bacterial load in combination with antibiotics. Now, after investigating all the action of these adjuvants through a number of in vitro studies, we wanted to check its toxicity in vivo. And we have checked the dermal toxicity by applying it on the skin of mice. And as can be seen from these histological images over here, even after the compound treatment of 200 mg per kg, the condition of the mice skin was similar to the saline treated control. And we have also evaluated systemic toxicity through a number of modes of administration like subcutaneous, intraperitoneal, and it could be seen that in this subcutaneous dose, the adjuvant was tolerated even up to 179 mg per kg. And we have also assessed the tolerated dose of the combination of minocycline and LB-DIFE by assessing certain liver function and kidney function parameters. And we observed that at a 30 plus 30 mg per kg dosage of both, it was tolerated by mice. And after evaluating its in vivo toxicity, we have further validated the efficacy of our combination in a P. aeruginosa skin infection model. And you can see over here that there was around 2.5 log reduction in the presence of the combination at 40 mg per kg plus 40 mg per kg of adjuvant antibiotic. And even in the individual treatments, no such reduction was seen. And this was comparable and even slightly better to the known gram-negative antibiotic colistin. So in conclusion, our combination exhibited efficacy in a murine skin infection model of P. aeruginosa. So finally, through our work, I would like to conclude, we have shown that class 1B adjuvants hold promise to be translated into the clinic. And we have developed an adjuvant which weakly perturbs the membrane of bacteria to allow more antibiotic influx and obviously less efflux, which results in bacterial cell death. And in this strategy, we have optimized the design parameters for such non-active, non-toxic adjuvants. And we have found out that cyclic hydrophobic moieties are a good resort to design such compounds. This showed potentiation against carbapenem-resistant strains of bacteria, which are the most critical priority pathogens. And further, we have improved our design and showed activity against more complicated forms of infection like biofilms and intracellular bacteria, and also proved efficacy in a murine skin infection model. But definitely there are some more things which need to be done, like we have to evaluate the efficacy of systemic infection model, like lung infections, urinary tract infections, etc. And obviously, because we are using this in a combination, the PK properties of the adjuvant and antibiotic to ensure sufficient bioavailability and resultant efficacy also need to be investigated. And we are currently working to achieve this. So with this, I would like to end my talk. Thank you very much for your kind attention, and I'm happy to take any questions.
Geetika, thank you very much for really an impressive talk. The floor is now open to questions. I think I was muted when I mentioned before, but please, if you're interested in asking a question, use the Q&A or chat functions, and then we will ask them. So I'll start. I have a few questions for you just to begin. The first one is from Josh Jackman, and he found your talk very interesting. He wanted to know, are the cationic amphiphiles active as individual molecules or as self-assembled nanostructures? That's the first question.
So no, so that was the idea of designing the adjuvant. We wanted it to be non-active and non-toxic in general. So the compounds are not active on their own, even at high concentrations of 512 microgram per ml.
Okay,
and,
so that was not, no, so the question is, do the compounds self-assemble to form larger aggregates or nanostructures? As opposed to acting as individual small molecules?
Okay, so actually the aggregation properties we have not studied yet, but that's a definitely a good point to look at it. So we have not investigated the aggregation properties of the compounds, but even at high concentrations of 512 microgram per ml, I can say that they are not active, like as individual molecules. But yeah, yes, the aggregation we have to investigate. Yeah.
Yeah, well, it wasn't aggregation. It could be that it's actually forming self-assembled nanostructures that are well-defined. Okay. And so not a non-specific aggregate, but there are molecules that do that with these amphiphilic properties, and I think it might be interesting to look at.
Yes, definitely.
Okay, so other questions for you. Let's see, chat, looking through here. I had written this few down here. Yeah, so I guess you haven't done any PK to determine, the viability of your molecules. You've really done a lot of work, but I think, what are your foreseeable plans, to examine how these operate in vivo? Are they going to be soon?
Hello, can you please repeat the question? There was
so the question is, you mentioned you plan to do some pharmacokinetic experiments to determine
yes, definitely.
That's really important because these molecules, the problem is that it's very difficult to tune pharmacokinetics. So if they have intrinsically bad properties, you won't be able to move them forward.
Right.
So do you have any idea? Go ahead.
So we are we are in the process of we still have to evaluate the PK properties of the adjuvant, definitely. But Yeah, and if like, say, for example, we have certain combinations I showed one adjuvant SPR 741, which are 2 different components, which have been used in combination to tackle infections, and they have entered clinical trials also. So that holds some promise for us. But in case like basically to decide the dosage and all, we need to do PK properties, and that would decide the dosing of our properties. And
Yes.
definitely like, yeah.
Go ahead. Yeah, so I have a question, another follow-up question. This is this is from Felix Calderon. He's an editor at ACS Infectious Diseases, and he said, thank you for the great presentation. He was wondering if you examined the development of resistance of your adjuvants. And so
Yes.
it certainly can get resistance to an adjuvant. Do you have any idea of the
Yes.
frequency of resistance or mechanism?
Yes, yes. So the frequency of resistance actually we have not evaluated, but we did passaging by treating for around 30 days by treating the same bacteria which were treated with the combination, and then treating it again with our combination. And we observed that the bacteria did develop some level of resistance. But when we took those resistant bacteria and we used the same adjuvant and changed the antibiotic, it was able to kill the bacteria. So indeed, that means that the resistance which was acquired was due to the antibiotic and not the adjuvant. So like that is the idea to our adjuvant, because it is non-antibacterial in nature. There is less pressure on bacteria to develop resistance just against the adjuvant. Mm-hmm. But we have observed that the resistance is against the antibiotic, and by choosing an antibiotic which has like delayed resistance, or by changing antibiotics, we can solve that issue. So yeah, that's my
great. Another question is, polymyxin resistance is due to remodeling of the cell wall to mask to change the charge from negative to positive. Right. And so that's not widespread, but it is concerning. And your molecules also, that would, I presume, confer resistance to your adjuvants. Have you thought about that?
So that's a good point. And to address this, we will take some colistin resistant strains, like some bacteria which harbor the MCR gene, like whatever you are saying, against which have the modified membrane where the charge is slightly from the negative to zwitterionic. So we'll take those bacteria and evaluate activity of our combinations against them. So that's a good point to look at, and we'll definitely do that.
Yeah, that's great. You mentioned host pathogen interactions when presenting alternate strategies to overcome antimicrobial resistance. What's your opinion of these other approaches?
Sorry, what?
Oh, so your introduction, I guess it was class 2
yes.
on targeting host pathogen interactions, really the host.
Right.
What is your opinion on those strategies?
Yeah, so those strategies also hold promise, and even for our adjuvants, we are looking at certain class 2 kind of interactions, because these molecules are positively charged and they can interact, like I said, with certain negatively charged enterotoxin in gram-negative bacteria, like like LPS. And LPS is known to cause inflammation in infections. And usually at that point of time, we require certain immunomodulators which can change the native form of LPS so that the inflammation is not triggered. So we are trying to look at the immunomodulatory potential of our adjuvants as well. And definitely the class 2 kind of molecules, they hold promise, and we'll look at that for our adjuvants as well.
Great. And I have a last question for you from Nathaniel Martin. He wanted to know if this synergistic effects of your adjuvants are antagonized by incubation with LPS.
No, so they are not.
So you did that experiment?
Yeah, I have done LPS, like certain experiments with LPS, like at in molar excess of LPS. So in presence of LPS, they are not antagonized, but we have also done the same studies in presence of certain divalent cations like magnesium, which are known to assemble LPS in the outer membrane of bacteria. So in the presence of magnesium, we observed that there was certain antagonization. There was a 64 to 128 fold reduction in the activity of the combination. So we presume that the interaction of our adjuvants with the LPS is like not very strong. It's slightly transient, which is probably not seen when we add excess molar of LPS. But when we add the divalent cations, they lead to the reorganization of LPS again, which antagonize our antibiotics. So yeah, we observed that.
Awesome. Well, Geetika, thank you very much. That was just an incredible talk. It was good.
Thank
Woke
you.
me up in the morning. So thank you.
Thank you.
Hold on. We'll have maybe open the floor at the end if there's further questions at the final end after Mangal talks, then we maybe return. But in the meantime, I would like to thank you, and Mangal, Singh, Dr. Singh, I've turned the floor over to you.
Yes, Courtney. Thank you. Shall I start?
Yes, please.
Sharing my screen. Yes. Thank you very much, Courtney. So I hope everyone can see my slides.
Yes, we can see them. They look great.
Yeah,
Tell us,
so,
you can begin.
hello, everyone. Today I'll be talking about a very interesting topic on development of beta-lactamase inhibitors for targeting carbapenem-resistant bacterial pathogens. So Geetika has already given a background about the antibiotic resistance and how big it is a problem. And in 2017, WHO came up with a priority list of the pathogens which poses the most threat to human health. And in this list, we see that most of these bacterial pathogens are gram-negative, and they are mostly resistant to one or different kind of beta-lactam antibiotic. And at the top of the list is the carbapenem-resistant bacterial pathogens, including the carbapenem-resistant Enterobacteriaceae. So these critical pathogens, they affect the people who need support of the ventilators, or they need blood catheters as a part of the treatment. So as per the CDC data of 2017 and the Indian Council of Medical Research annual report of 2020, we see an increasing trend of carbapenem-resistant Enterobacteriaceae. So in 2020, the number of isolates collected by the ICMR in different labs around India was affected by COVID-19. And compared to 100,000 samples collected in the previous year, only 65,000 samples were collected. And the top pathogens that were isolated were Escherichia coli and Klebsiella pneumoniae. And more than 50% of these Enterobacteriaceae were carbapenem resistant. So the question is why carbapenem resistance is so much of a concern. So we see carbapenems are last resort beta-lactam antibiotics. What it means is that they are prescribed to patients when the previous generation of beta-lactam antibiotics don't work, like when the penicillins, cephalosporins, and monobactams become ineffective. So then now even resistance to carbapenem has jeopardized the solutions to deal with these pathogens. And bacteria has evolved a different mechanism to resist the action of these beta-lactam antibiotics. So to resist the action of carbapenems, the last resort antibiotics, the bacteria has evolved different mechanisms. So what happens is that beta-lactam antibiotics, they reach the periplasmic space and target the penicillin binding proteins, which are involved in biosynthesis of cell wall. So what bacteria has evolved is it has evolved beta-lactamases, which are either genome encoded or plasmid encoded, which chop this antibiotic before it can target the penicillin binding proteins. The bacteria has evolved efflux pumps, which can actively efflux out and reduce the concentration of the beta-lactam antibiotics in the periplasm. And the channels which are the passage for these beta-lactam antibiotics to reach the periplasm are modified or underexpressed. So these are the different mechanisms by which beta-lactam or carbapenem resistance is achieved by the bacteria. So the main mechanism that is involved in carbapenem resistance is production of the enzymes known as carbapenemases, which hydrolyze these. And they are broadly classified into serine beta-lactamases and metallo-beta-lactamases. So the serine beta-lactamases have an active site serine residue, which opens up this beta-lactam ring, thereby making the antibiotic ineffective. In case of metallo-beta-lactamases, there is no active site serine, but instead there are zinc ions which activate a hydroxylate, and this hydroxylate does a nucleophilic attack on the beta-lactam ring to open up and thereby making the antibiotic ineffective. So the major carbapenemases of concern are the carbapenamase KPC2, the class D OXA48, and the metallo-beta-lactamase NDM1. So I request Lorraine to ask the first question, and then we can continue.
Okay, so we have another poll question for the audience that I will pull up now. Okay, and the question is, which of the following beta-lactamases exhibit broadest range of substrate hydrolysis, including that of penicillins, cephalosporins, monobactams, and carbapenems? Is it class A, class B, class C, or class D? We'll give everyone a few seconds to respond. And I see there seems to be a runaway answer. We'll give everyone maybe 10 more seconds. Okay, and I'm going to close the poll in 3, 2, 1. And here are the results. Back to you, Mangal.
Okay, so most people have answered NDM1, and so NDM1 and OXA48 cannot hydrolyze the monobactams. So it's the KPC2 which can hydrolyze the penicillins, cephalosporins, monobactams, and carbapenems. And MC is not a carbapenem, so that's not the correct answer. So yes, there can be a discussion on KPC2 and NDM1, which is which hydrolyzes the broadest. But if we see the 4 classes, it's the KPC2 which hydrolyzes all the beta-lactam antibiotics. So so moving on, so we in our study targeted KPC2, which is carbapenem of concern because it can hydrolyze all the 4 classes that I initially showed. So we started with the previous study where the cell enzyme group, what they did is they took a serine protease, modified it, and added a carboxyvinyl arm to it, and found that this addition made this serine protease a beta-lactamase inhibitor, and the potency of inhibition was very much improved. So they co-crystallized this and found that the molecule covalently targets the active site serine residue and makes multiple interaction at the active site. So what we did is we did a virtual analysis using a software which allows modification in the molecule virtually, and by taking the help of this CSAR analysis software, we designed 17 derivatives from this previous work. So 15 of these molecules were acyclic and 2 were cyclic. So then we wanted to test whether these modifications did any improvement to the original molecule. So what we did is we purified these 2 carbapenemases and we checked the half maximal inhibitory concentration of these molecules. What we found is that most of these molecules inhibited KPC2 and the carbapenemase OXA48, and both were serine beta-lactamases. We also checked whether these molecules can inhibit the metallo-beta-lactamase, and as there is no active site serine residue, so no inhibition was observed. So as these molecules were derived from a molecule which was originally a serine protease inhibitor, we wanted to check whether these modifications are responsible for specific beta-lactamase inhibition, or they can also inhibit a serine protease. And what we found is that the molecules cannot target the serine protease trypsin. So these molecules were quite specific to the serine beta-lactamases, and that is KPC2 and OXA48. So then next we wanted to check whether these molecules can work on a clinical strain. And for that we took a KPC2 producer, Klebsiella pneumoniae strain K4725. This strain was isolated from sputum of a patient admitted in a Norwegian hospital who had a history of administration in a hospital in Greece. And this particular bacteria produces the carbapenemase KPC2 and the beta-lactamase SHV and TEM1. There are 2 copies of beta-lactamase in it. So from the table, we can see that this particular bacteria is resistant to the penicillins, the different generation of cephalosporins, and the cephalosporin classical beta-lactamase inhibitor combinations, as well as the monobactam and the different carbapenems. So we tested its MIC and we found that this particular strain, it comes under resistant range. That is, it shows an MIC above the CLSI breakpoint. And when combined with the parent molecule that we started with, we see that the bacteria becomes sensitive to the action of meropenem. And with the different combinations, we see that some of the molecules reduce the MIC of meropenem even further. That means they are potentiating the activity of meropenem. So what is happening here is that some of the modifications enhance the potency of the molecule from the original molecule. So what happens is, in the resistant bacteria, the meropenem gets entered through the porins. The expressed carbapenem hydrolyzes this antibiotic before it reaches its target's penicillin binding proteins, and hence shows an MIC of MIC that classifies it as a resistant strain. But when combined with these inhibitors, the inhibitors block the action of the carbapenemase and thereby allowing meropenem to target its specific penicillin binding proteins, causing spheroblast formation and reduction of the meropenem MIC. So the carbapenem inhibitor combination has reduced the amount of meropenem required to kill this bacteria by more than 128-fold. So then we wanted to check whether this molecule shows synergy in a checkerboard assay, and we found that the molecule show a dose-dependent enhancement in the activity of meropenem. And in the time kill curve, what we found is that this particular molecule shows bactericidal activity, and the bactericidal activity was better than the commercially available beta-lactamase inhibitor, that is Weber-Bactam. So what we found is that these molecules alone are non-inhibitory, but when combined with meropenem, they reduce the meropenem MIC several folds, and they potentiate the action of the carbapenems. So we come to the next question, and I request Lorraine to answer, please.
All right, thank you. So we have one more poll question for the audience, which is coming up shortly. Okay, it is based on the data presented up to the slide, which other carbapenemase producing bacteria need to be tested for the potency of these lead molecules? So is it a KPC2 producer, an NDM1 producer, an AMP-C producer, or an OXA48 producer? Okay, and we have the results starting to come in. No runaway answer just yet. Okay, we'll give everyone a little bit longer to answer, and we'll close the poll in about 10 seconds. All right, I'm going to close the poll in 3, 2, 1. And here are the results. Back to you.
So yeah, so most people got it right, but there are I see equal number of answers for all the options. So as I said in the beginning, that these inhibitors mainly target the serine beta-lactamases, and we were getting activity against the serine beta-lactamase, the KPC2 and OXA48 in the enzymatic activity assays. So the next bacteria that we need to test is an OXA48 producer. So for that, we took another clinical strain, and this clinical strain was a urine isolate from a patient who was admitted in a Norwegian hospital, and this patient had an history of travel from Thailand. So this particular bacteria produces the carbapenemase OXA48, CTXM group 1 beta-lactamases, and OXA1 group beta-lactamases. So we see that this bacteria is also resistant to the action of cephalosporins, the penicillins, and this bacteria is sensitive to meropenem. So yet we wanted to test whether any of these molecules which show in vitro inhibition of OXA48 activity will show any reversal. And what we found is that only one of these molecules restored the meropenem MIC. So there was an 8-fold reduction in the MIC of meropenem against this particular strain. So out of all the molecules, only 2 molecules were cyclic, and only 1 molecule was found to be potentiating the activity of meropenem. In the checkerboard assay, what we found is that the molecule helps to reduce the meropenem MIC in a dose-dependent manner. So next, we wanted to check whether the binding of this inhibitor is stable or the enzyme revert back as soon as it is diluted or the inhibitors are removed. So what we found is that the parent molecule, which we started from, doesn't show a stable binding, and as soon as the inhibitors are removed, the enzyme recovers completely as if there was no inhibitor. But with some of the molecules which are highlighted in yellow, it takes some time for the enzyme to recover, and we calculated the residence time of these inhibitors on the KPC2. So we see that out of all these molecules, some of these molecules show a residence time of approximately 108 minutes. That means they hold on to the enzyme. But what was surprising is that none of the OXA48 inhibitor, which also showed a reversal of the meropenem MIC, was a stable binding inhibitor. So next, we wanted to check one of the molecules which showed all the activities, good activities against the capacity producer. So which we checked the toxicity of these molecules against peripheral blood mononuclear cells, and we also checked their hemolytic activity. What we found is that none of these molecules were hemolytic, and they had a CC50 value of more than 200 micromolar. So they exhibit a non-cytotoxic behavior, that is more than 95% cell viability was observed even after incubation of 24 hours. So based on our data, our cytotoxic data and a previous report which says that boron containing compounds are safe to administer to mice, and they exhibit an LD50 of more than 900 mg per kg, we thought of doing efficacy analysis in a mice pneumonia model of infection. So we infected mice with a strain which was very much resistant to most of the beta-lactam antibiotics along with the carbapenems. And what we observed is that the monotherapy was not able to reduce, monotherapy of either the molecule or meropenem was not able to reduce the bacterial load in the lungs. But when given in the combination, the combination led to more than 0.9 log reduction of the bacterial count. So this indicated that in combination with meropenem, the molecule works in a biological system and the inhibitor is functional. So to summarize, we started with a known KPC2 inhibitor. We made 17 of its derivatives. And what we found is that 7 of these molecules displayed improved activity over the original molecule. And one of the molecule also exhibited OXA48 inhibition in addition to the KPC2 inhibition, which was not actually present in the original molecule. So we checked the efficacy and found that the molecule is efficacious in reduction of the bacterial burden, and we obtained multiple beta-lactamase inhibitor. So this work was a part of an Indo-Norway project, and it was a collaboration between Indian Institute of Technology, Roorkee, and University of Tromsø. Mark Boomgaren, who's a postdoctoral fellow in Professor Annette Weir's lab, synthesized these molecules. Professor Henna provided us with the clinical strain and constructs for the enzymes. This work was carried out in our lab at IIT Roorkee. So Professor Ranjana Patani is the supervisor. Pravez Bhatt and me did the biological work. And I would like to thank ACS for providing me this platform to discuss the progress of this work. So thank you for your attention. So I would like to take any questions.
Yes, Mangal, thank you very much for an exciting talk to finish up our session. The floor is open to questions, so please type your question in either the chat or the Q&A.
Yeah, I could ask a question, Courtney.
Go ahead.
It's a very nice talk, Mangal. I was wondering whether you you measured the post-antibiotic effect of any of these compounds.
Yes, so I did that experiment, but not included here. So when combined with the these molecules, the meropenem shows 1 hour delay, like 1 hour post antibiotic effect, approximately one hour of post antibiotic effect.
That's in the presence of your boronic acid.
Yes,
inhibitors?
when compared to meropenem alone.
Oh, very interesting. And does the parent boronic acid also have a residence time on the enzyme?
Yes. So they have almost similar post-antibiotic effect. So no doubt on that.
I had a question, an enzymology question, just on your IC50 values you reported. What is the enzyme concentration used?
Enzyme concentration was less than 1 nanomolar. It was approximately 1 nanomolar.
Okay, so there's no tight binding behavior being observed.
What kind of behavior? I didn't get it.
Tight binding, so if your IC50 is close to the enzyme concentration, then it may indeed be more potent.
Yes.
Another general question is, so these, what do you call this class of molecules? The boronates are boronate beta-lactamase inhibitors have become
phenylboronic acid based
fairly
inhibitors.
popular, and so we have Vaborbactam was approved several years ago.
Yes.
And there's 2 preclinical candidates. I don't know if you've seen in the literature, Tanorbactam by
QPX7728
and QPX7728 by QPX Pharma in San Diego. And both these are very, very advanced, and you didn't comment upon those or use any of those as controls in your assays. And I think it would be nice to include some other comparative controls in discussing your inhibitors.
Yeah. So I did not included the Taniborbactam and the QPX7728 molecule. So both are broad spectrum carbapenem-resistant inhibitor. They target both serine beta-lactamase and beta-lactamase. But I used Vaborbactam, which was available to us as a control. Yes.
Okay. Yes. Yeah. And so do you see any limitations for your molecules? Do you see any advantage, compared to some of those other ones in terms of any properties or spectrum or potency that?
So what I see in the time scale kinetics is that the combination of boronic acids with meropenem is bactericidal within 2 hours, while with Vaborbactam it takes a longer time. Plus, for this strain, we see that at the same concentration, if we compare the equimolar concentration of Vaborbactam and these molecules, for this particular strain, the boronates work better than the Vaborbactam in reducing their MIC. So
yeah, that's great. Yeah,
yes.
That's very nice. I see no other questions coming in. I have, let's see, I'm gonna checking the chat more, see if we have any more. Okay, last question from Felix is, what do you think are the next step for your compounds?
So we see that one molecule here, which we started as a KPC2 inhibitor, turned out to be an OXA48 inhibitor. So we'll be comparing it with Vaborbactam and the molecule that we tested in this particular study. So this molecule turns out to be an inhibitor of KPC2 OXA48, and as this is an ongoing work in the lab, we see that this molecule also inhibits the class C, MC, and the extended spectrum beta-lactamase, CTXM1. Except for metallo-beta-lactamase, it inhibits all the 3 classes of beta-lactamases, including the carbapenem-resistant. So we'll be doing animal studies and we'll be further developing this particular molecule, which seems promising because it is showing broad spectrum inhibition.
Yes, wonderful. Thank you very much. So,
Thank you,
Lorraine, any final comments?
I just want to thank everybody for joining today. Thank you also to the audience for providing some great questions and for the interaction with the speakers. I also want to thank Geetika and Mangal for joining the session today as speakers. And of course, many thanks as well to Courtney and Pete for moderating. Thank you, everyone. Have a great day.
Thank you.